So thank you again to Dr. Rem. I'm delighted to introduce the moderator for our next panel, Dr. Shamil Sunyaev. Dr. Sunyaev is a professor of biomedical informatics at Harvard Medical School and of medicine at Brigham and Women's Hospital. He's a leading computational genomics and genetics researcher, and he's been a pioneer in computational and statistical methods development. These include household names for many of us, like PolyPhen. That have been widely adopted by the research community and in clinical grade variant classification algorithms. So Dr. Sunyaev, thank you for joining us, and please take it away.
Thank you very much. So we have a discussion panel on polygenic risk scores and why aren't they adopted widely. So I'm extraordinarily happy about the composition of the panel. I'm happy to introduce the panel. So first we have Alicia Martin. Alicia is a faculty at Mass General Hospital here and at Broad. So Alicia started studying human evolution and issues of human population diversity, doing PhD with another panel member to be mentioned later. And then she moved to Boston to a more medical genetics environment. And her research took a turn into studying biases that are brought to medical genetics from insufficient consideration of population diversity. And now her own research program focuses on these biases and how to deal with these biases. And maybe the most known work is on the issues of transferability of polygenic risk scores. So Alicia showed that they are poorly transferable between populations and that this is a phenomenon related to demographic history rather than differential biology. So we're very happy to have Alicia to contribute to this. Then Anirudh Patel is, fortunately we have a clinician on the panel. So Anirudh would provide clinical perspective. He's a clinician at Massachusetts General Hospital attending, but also a faculty member, research faculty member, early stage mentored investigator. His focus is on interpretation of variants, which is relevant to the rest of this meeting, and on predicting cardiovascular disease based on polygenic component and also epidemiological non-genetic factors. So he publishes in these areas, and I think his own research will be placed solidly in predicting risk, understanding risk, and employing risk of cardiovascular disease. So then we have our currently representative from industry, Stella Avdikian, and she is a researcher at 23andMe, interested in multiple applications in application of prediction, right? The disease risk from both genetic and non-genetic factors. She contributes to studies on specific disease areas like Parkinson's disease. She participated in the COVID study by 23andMe. And of course, she's involved in the drug discovery effort. So as she told me, she's jack of all trades, or maybe all trades, collected by 23andMe. In her previous academic life, she was studying genetic epidemiology of cardiovascular disease, but now, as you see, her industrial portfolio is a lot broader. And then I'm amazed to have to introduce Carlos Bustamante as a panel member. And Carlos, maybe if you probably know Carlos, Carlos is a leading population geneticist who I don't know if he agrees, but I think the main contribution is that with very solid population genetics background that he got doing his PhD in this town, I believe, well, across the river, he brought this theoretical background to the analysis of data. So he connected with Bayesian methods and many inference techniques. He connected population genetics diffusion theory to massive data on genetic variation, and that's an amazing contribution to the field. He also represents multiple, with exception of clinic, right? So Anirudh is the only clinician on the panel, but he represents different areas of interest, right? He, as academic researcher, he spent his time at Cornell, being faculty there and being a faculty and then inaugural chair at Stanford, and I believe still maintains the appointment at Stanford. And then he's also involved in industry in both sides as venture side of the industry, working with F Prime, the company that invests in biomedical endeavor, and working with data science in health there, and also is advisor to multiple companies, and now as an entrepreneur starting a company Galatea, which has a lot of relevance to what we're doing today. So I'm very excited to have the panel. We decided to collect one fun fact, just happened, that Carlos's favorite city is Nashville, right, Carlos?
That's right.
For Stella, surprise, surprise, Yerevan, Armenia is the favorite city. Then for Anirudh, interestingly, favorite city is Berlin, and for Alicia, the favorite city is Seattle. So this is introduction of the panel. I will say maybe just a few words setting up the question, and we'll start with panel providing their responses. Some of the responses I've seen in writing, some of the responses I will hear for the first time, and then we'll field questions from the audience. So the broad issue here is, what are the hurdles, what are the barriers in front of broad introduction of polygenic risk scores into the clinic, into industry, into many areas, many practical areas of human genetics, right? So first, what are polygenic risk scores? Most of this meeting is dealing with rare disease, Mendelian disease, where a signal variant has profound effect on disease risk, right? So this variant carries a lot of information, whether the individual would develop or not develop the condition. In most common diseases, mostly of interest to medicine, that's not the story. The story is that we have highly polygenic architecture, meaning very many variants with tiny effect sizes, meaning that the information about any specific variant carries almost no information. So in Mendelian genetics, you can predict initially from pedigrees, from segregation, and now from actual genotyping, you can predict risk. In common disease, you still can predict risk. If one allele has no information, it doesn't mean that looking at them collectively, right, at the combination of independent small effects, this carries the information. And the first polygenic risk models were developed in the 1970s, again, looking at what I was told, heard of the state of New York in cattle, and then moving to direct genotyping applications. And they demonstrated incredible success in agriculture, right? Agriculture relies on these models. And then as early as 2007, to my knowledge, there is first theoretical paper on potential application in humans. 2009, first application of concept of polygenic scores into humans. And this has been a playground mostly for statistical geneticists, for more theoretically minded community. And a few years, maybe 3-4 years ago, the discussion about, led by Sekathrezn and others, the discussion started whether this is already good enough to be applied in practice to evaluate risk of human common disease. Now, the question in front of this panel is, why in the world we're still not applying those methods widely, right? We apply genetic risk in monogenic sense in clinic a lot. Why not polygenic risk of common disease? And I wanted specifically to go a little bit with the questions for the panel, go a little bit more specific. So first, obviously, we all want these models to be more accurate. How can we make it more accurate? Is it new mathematical models, new amazing mathematics? Is it collection of larger and larger samples? Is it feasible for diseases that are not very common? So we'll need disease-specific panels. So what's the best way forward to make the models more accurate? Second, again, as discussed when I introduced Alicia and Carlos, transferability between populations. Huge hurdle. You cannot have a clinical method only applicable to one population where you have data from. And that's an amazing problem. Again, is the solution with mathematics? Is the solution with broad sampling of human diversity? Or maybe solution is with biology, because the whole problem is that we're dealing with markers. We're not finding variants causally implicated in traits, and this is what causes this issue, right? So maybe the solution is biology, mathematics, or data collection, or biology. The next one, so beyond these fundamental issues, what are barriers within the healthcare system? Because obviously there is resistance, right? Not fully explained by a conceptual scientific problem, and Anirudh and Stella probably would comment on this. And the very last point is, okay, if we're not ready to apply it immediately in the diagnostic setting, in industry, drug discovery, stratification of patients, right, running trials, and I think maybe I know Stella is involved in drug discovery. Maybe she can comment, or maybe others as well. And I think Carlos might comment. What are the uses that are practical uses of polygenic risk, but maybe not directly as a diagnostic tool? So I think I said enough about posing the questions, and we'll start. I think each panel member maybe 3-5 minutes of your thoughts. You can answer all questions, some questions you can answer something I didn't ask. So Alicia, please.
Sure. So I think to kick us off where you started, Shamil, the question of whether mathematical models are going to take us there, whether data is going to take us there, whether biology is going to take us there, and there being clinical translation of these polygenic scores. I think as a field, we're pretty good at walking and chewing gum simultaneously, actually. So the carry forward of all of the mathematical models are based on like a century's worth of theoretical framing. These mathematical models that polygenic scores are based on have moved really incredibly quickly in terms of their development and their applications in human populations. And so that's been really exciting to see. There's just been this deluge of methodology development. The second part of that, though, is harder. So I see the single greatest limitation to genetics in precision medicine for common diseases as the fact that our data sets are so vastly Eurocentric. So just a few numbers on that. Where are we coming from? Well, the first draft of the human genome sequence was published in 2001, cost $2.7 billion. Now you can routinely get a human genome sequence for like $600, and an estimated 30 million people have been sequenced. It's even cheaper to produce the data needed for a polygenic score. So it costs like $50 a sample to produce the data that goes into producing a polygenic score, which is incredible and means that's cheaper than a lot of our clinical tests that are being routinely used in medicine right now. So that's phenomenal progress over the course of the past couple decades that suggests that we should really rapidly be approaching translation for common diseases, which the vast majority of people are going to experience at some point in time. But what I see as a huge problem is there's a study in 2009 showing that 96% of participants in our genome-wide association studies are of European descent. And a decade later, we published on this same problem and showed that now 80% of participants in genome-wide association studies are of European descent. So over the course of a decade, when there's this vast explosion of data, we're still not anywhere near where we need to be if we're trying to reflect national or global representation, and the US government is one of the largest funders of biomedical research in the world. And so who are we actually representing? Clearly we're not representing the US population. We're not representing the global population by any means. Only about 16% of the world's population is of European descent. What we're really good at representing is the makeup of academic researchers, in fact. So in order to actually push this forward, we need to sort of democratize genomics, I think, in a lot of ways. So a lot of government researchers, a lot of academics, and a lot of companies are actually driving forward, changing the composition of our genetic studies. But it doesn't happen overnight, and there's a lot of reasons for that. There's a lot of mistrust. There's a lot of differences in terms of who has access to healthcare. In minority populations in particular, this is a huge challenge. All of our established researchers and all of our established consortia are primarily, building off of these decades worth of research, and they've already sort of got these entrenched and ingrained practices. There's statistical reasons to try to reduce false positives in our genetic studies and removing ancestry outliers. It just means we keep propagating a lot of these study biases. So to give sort of an example of what I think has been like a really exciting project that we've been able to work on is the Neuropsychiatric Genetics in African Populations project. So this is a project set up to study psychosis, schizophrenia, and bipolar disorder. We've set up this study in 2015 across 4 countries in Eastern and Southern Africa: Kenya, Ethiopia, Uganda, and South Africa. The goal was to collect 40,000 participants, and remarkably, we've been totally on target with all of our recruitment goals, but it literally took until this year, so 7 years after we started the project, to be able to collect those 40,000 samples. This is an enormous data set, a scale that's not been seen in many of these countries participating in this study. So why has it taken so long to get this stuff off the ground? Well, there's IRBs and ethic reviews committees that are not familiar with evaluating the benefits of genomic studies or what the potential harms could be to communities involved. There's a lot of mistrust, as I mentioned. There's training the clinical workforce. There's building a lot of the infrastructure that Heidi Rehm was talking about earlier. And then there's making sure that the labs are conducting the work in a similar way. So there's a lot of harmonization and protocol development that needs to happen. There's the need to do like really technical things like minimize sample swaps, minimize contamination. And then there's training the researchers. Really, the people power is all there. There's incredible researchers everywhere around the world. But conducting the cutting edge analysis also requires years worth of training to be able to lead a lot of that work. So I think there's just an enormous amount of work that needs to be done to sort of set up these issues on the data collection side. And then translating polygenic scores really requires also, as you mentioned, Shamil, some development in what is actually the causal biology. Polygenic scores are just tagging correlations, which means we're not using causal biology to build these predictors, which are kind of akin to biomarkers that are already being used in clinical medicine. So if we're just using correlations and we've got this big complicated population history that's driving a lot of the scores, then we also see that there's a lot of biases that trickle into our polygenic scores. So when we try to translate these, there's going to be some big issues that are just going to come right back to how humans populated the globe, and nothing to do with anyone's health status that impacts how people are going to understand accuracy and how risk is going to be conveyed. And I think this is the issue of polygenic scores in the clinic is like really challenging, and I'm glad that we have a panelist who can actually speak to that, because understanding the clinical side is really not my forte. But just communicating risk when there's biases that are not like other kinds of risk factors that people are really used to dealing with, like smoking and hypertension and BMI for cardiovascular disease. The polygenic score is as predictive in European ancestry populations as any of those factors right now. It's much less so as you get more genetically distant, but understanding how to communicate that kind of risk, that it's not just because of your health differences that you might see differences in prediction accuracy, but it's actually just because of who your ancestors are. And I don't mean like your parents or your grandparents, I mean like your ancestors throughout all of human history over the course of the past hundreds of thousands of years impacts how we might convey this. So it's a big old hairball that is a little complicated, and I think other panelists are better suited to commenting on the clinical risks.
Right, so Anirudh probably will tell us how the healthcare industry would deal with hairballs. So.
Yeah, so I'm thankful to be a part of this panel and bring the clinical perspective. So as Dr. Sunyaev introduced, I'm a cardiologist. And from the perspective of cardiology, we have a lot of, a wealth of data from clinical trials that tells us how to manage patients who develop heart attacks and other cardiovascular diseases after they happen. But as a researcher, my goal is to design better tools so that we can predict this disease and try to prevent it from occurring. And genetics provides a wonderful window to help improve our tools, from using basic things like LDL cholesterol and blood pressure to incorporating rare variants to incorporating polygenic variants. And the traditional model in terms of how we use genetics in cardiovascular clinic has largely focused on these rare Mendelian disorders, so things like familial hypercholesterolemia, where a handful of variants and a handful of genes lead to higher circulating levels of LDL cholesterol, so that the carriers of those genes end up having heart attacks in their 20s and at a much higher frequency than the rest of the population. But as the panelists and Dr. Sunyaev have kind of described, that polygenic risk scores are able to capture all the minuscule effects of all the variants across the genome into one single score. And our group within Mass General Brigham has done a lot of research in terms of trying to translate that score into clinical applications. And some of the reasons why the polygenic score in clinical practice excites me are that, so for one, you can quantify genetic risk for a number of diseases for an individual starting at birth. and that gives you decades to try to help prevent those diseases. For cardiovascular disease in particular, the top 8 percentiles of the polygenic risk score for CAD identifies individuals who are at 3 times the risk of developing a heart attack compared to the rest of the population. And so that's a remarkable relative risk. And that's actually equivalent to what someone with a familial hypercholesterolemia variant would experience relative to the rest of the population. Similarly, risk prediction using these polygenic risk scores exceeds that of any of the traditional risk factors that we use, like blood pressure, cholesterol, either individually or with all those traditional risk factors combined, polygenic risk scores still come out on top. Similarly, polygenic risk scores identify people at risk orthogonally to just using the risk scores alone or even using family history. If we were just using our current method, we'd be missing so many people who are at high polygenic risk because it provides additional information. And among the people that are identified to have high polygenic risk, DNA isn't destiny. Observational studies have shown that improved lifestyle or statin or cholesterol-lowering medications can help decrease both the relative and absolute risk in individuals with high polygenic risk. So with all these things that are going for polygenic scores, so why aren't they kind of being used by everybody and everywhere? And I think there's several kind of problems or hurdles that we need to overcome. So Dr. Rem described phenomenal work, particularly within the rare variant space, where many different stakeholders and many different parts of medicine came together to try to optimize care for patients who are presenting with rare disorders. But when you think about polygenic risk score, that's basically everybody is a candidate. So it goes from being about 1 or 2% of the population to 100% of the population, where we have to design some sort of assay for, some sort of protocol for evaluating, and then giving guidance on how to manage that risk. So just that scale alone is a big deal, regardless of how much data we have to say that it's a good idea. And so one of the issues is the issues of portability that Dr. Martin described. And just simply put, it's not fair to offer a test that only works for a certain percent of the population, and for the rest, even with a caveat, say, okay, this may, we're giving you a number, it may not be accurate. So in medicine, there's like a slightly higher bar if you're actually practicing and giving recommendations that they apply to everybody. So I think the portability issue is a major one that we can address as a panel. Secondarily, and how do you accurately and comprehensively report these findings, both to a lay audience like the patients, as well as clinicians who haven't heard of what polygenic risk scores are. So I graduated med school in 2014. I hadn't heard of the term polygenic risk score until I started doing research in it, over the past 5 to 6 years. So, the vast majority of primary care physicians who would be essentially the ones dealing with these reports that are coming in need to be educated as a workforce. And as far as educating the general populace, I think, getting into ideas of probability instead of, you have X risk and you will get something. I think that's a complicated language that we need to develop better tools and better reporting to try to convey that risk. Finally, there's also logistical aspects, which, there's a whole boatload of those. So one of those being, if we're going to be doing these sorts of clinical grade assays, the laboratories that are running these platforms have to be equipped and standardized to be able to do that through CLIA designation. We have to be using the correct samples. We have to have appropriate storage. And even if we do calculate a polygenic risk score in somebody, that'll get revised as we get more data and better scores in the future. So we have to have infrastructure for that, and then ultimately conveying all those results and all the uncertainty that comes with that. So although we have all these things going for polygenic risk scores in terms of the potential, and I just talked about cardiovascular disease, there's so many other applications within the cancer space and other rare disease spaces. There's still a lot more work that needs to be done, both logistically and then, at the level of education to help overcome those hurdles.
Thank you. So we're moving to Stella. I think there is one interesting component in Stella's work, because everybody is looking at data, maybe UK Biobank, but I believe that Stella has seen how the scores perform in a much larger cohort. So maybe there is a perspective there as well.
Yeah, that's right. So the cohort that Dr. Sunyaev mentioned is the 23andMe customer cohort, and we're talking about a really gargantuan one. I mean, at this point, we have more than 11 million genomes, And in addition to the genomes, we also have rich phenotypic data, both self-reported as well as subjectively ascertained. And when you have this much data, both on the genetic and the phenotypic side, of course, what are you going to do? You're going to run GWAS. And once you have good GWAS, you can start constructing PRSs. And our mission overall is to help people access, benefit, and understand their own genetic heritage, their own genome. So we do that in a couple of different ways. The way that's probably most familiar to the audience is our consumer product. It's that stuff that tells you where your ancestors came from, but it also tells you a lot more, especially in the health space. It tells you a lot more about your disease risks, and that includes both things like the BRCA1 and 2 mutations, so well-known single gene polymorphisms, but also we are incorporating polygenic risk scores in our product. We have a subscription service where it's kind of like Netflix, but for PRSs, and perhaps not as engaging, but perhaps also more impactful. And every month, our subscription customers get a new report based on a polygenic risk score. So it could be something like anxiety. I believe that was our latest release. And we have now infrastructure that enables us to look at this giant cohort and to actually generate and validate those reports pretty quickly. Because we have 12 million people, in absolute numbers, we actually have pretty big cohorts of African-American individuals, of Latinx individuals, of different individuals belonging to different ethnic groups. Now, if you look at it as a percentage, 3/4 of our population is still of European descent, so we have a lot of work to do in terms of reaching out and building trust with the communities that had historically been hurt by medical research, and specifically genetic research, and that's going to be a long path for us, but we're committed to it. But even now, we can already generate PRSs that work pretty well across ethnic groups. And in fact, one of our criteria for releasing a PRS to our customers is equity. So if it performs poorly in at least one ethnic group, we don't release it at all because we don't want to perpetuate existing disparities that exist in that space. So that's one way in which we use PRSs. The other way, and here I have to be very careful because I really, really like my job and I don't want to break any intellectual property agreements. So let me speak more broadly about how PRSs can be used in drug discovery. And let's just say that 23andMe understands and appreciates the opportunities that are afforded by PRSs. And we do currently have a couple of drugs in trials. They're in the immuno-oncology space. So how can we use PRSs in drug discovery? There are really two ways that the FDA specifically talks about. They distinguish between prognostic and predictive use of those biomarkers. And to me, those distinctions are a little silly. Those two words seem to be synonymous, but they specifically define prognostic enrichment as a way to get more events in your cohorts. In other words, attract people with higher risk. Now, this could be genetic risk. This could be risk due to people's lifestyles. And because this cohort is enriched for high-risk people, you will get more events. Perhaps you can do the trial in fewer people, and the trial might end sooner, so you're saving time and money by having that kind of enrichment. You have to also be careful with that kind of enrichment. We have some examples from the Alzheimer's space, for example, where the baseline risk was so high that it's actually very hard for you to see any effect of the drug because the rate of events in both groups is so high. Now, the other way to do it would be through predictive enrichment, and this is using polygenic risk scores or any other risk stratification algorithms to identify subgroups where the treatment is likely to have outsized benefit. And this is especially relevant. There's been some success in doing that in the oncology space. Now, this all sounds very promising, but it's also important to remember that we currently don't really have good prospective evidence. And I'm an epidemiologist. I thrive in that cohort study evidence space, right? All we have now are some trials that have been analyzed post hoc in a way that those participants would be genotyped and then would go back in time and stratify them based on their polygenic risk and say, oh, actually, if we just look at the effect of this drug in the, for example, high PRS percentile, that drug effect looks differently. Maybe the drug's actually more effective for these people. And that, again, all looks very promising, but to this day, I'm not aware of a study that actually recruited people based on their polygenic risk and then followed them up and then prospectively ascertained the contribution of such risk stratification algorithms. Now, I think those studies are coming, and we're definitely seeing that with single SNPs. We're seeing that a lot in the oncology space yet again. And I think particularly in cardiovascular medicine, we are going to be seeing more such approaches in drug trials as well. But I still think this is more an area of promise rather than an area of accomplishment at this point. And I think there are a few limitations, and one of them Dr. Sunyaev here alluded to in the beginning. The biggest publicly available data set that everybody works with is the UK Biobank. Now, it's got two problems. It's about 500,000 people, and it's very, very homogeneous ethnically. It's predominantly white, much more so, for example, than even the 23andMe cohort. So you have limited portability of PRSs that come from the UK Biobank, and you also, this is something that we see in the 23andMe data set, just don't have enough people. You don't have enough people to generate good PRSs that capture a large portion of the disease or trait variation. So I think part of it is just access to data. You asked this question whether it's going to be data or mathematical tools or the biology. Well, as always, the answer is all of the above. But I do think that the data access issue, the data availability issue to generate good PRSs is not trivial, because the effects that we're trying to pick up are very, very small. So you need a lot of statistical power to be able to pick it up and aggregate them and then deliver a meaningful polygenic risk score. Heritability, I think, also plays a role. So not only are some diseases rare, but some diseases are not particularly heritable. So probably those are not ones where we should be investing a lot of time and money in polygenic risk scores. There's some interesting discussion about progression versus onset. So can we use the same polygenic risk score to predict how a person's disease will progress, as well as predict susceptibility to the initial disease onset? And we don't have a lot of good evidence to disentangle those two issues, but that's one that I think is very interesting. And finally, Dr. Patel, you alluded to that. We don't know how polygenic risk interacts with other factors, for example, lifestyle, and we've started the work trying to disentangle them because we actually have a lot of lifestyle data on our customers, including data generated from wearable devices. So those would be really good objectively ascertained data. And we're going to be presenting some of that at ASHG, so we have a poster, come see it. And on that shameless plug, I will end with just one last caveat that, of course, equity is a huge problem that I think also affects the industry, but two of my colleagues here are much more qualified to talk about that.
Thank you very much. And I think for the discussions, I'm really interested in your comment that prospective studies are needed, and I know that many statisticians believe that in this setting they are not really needed, so that's an interesting point of debate.
I'll be happy to talk to that. Let me just briefly say that they're not wrong, because in a sense, any study of a PRS is prospective in nature because our genetic variation is established.
That's exactly the
conception.
argument. That's
Right.
exactly the argument.
And I think that's a really valid argument. Nevertheless, especially if we're integrating other clinical risk factors that are non-genetic, this is where you do need to worry about such things.
Thank you. Carlos.
Thank you. First, I just want to thank all the other participants. I've learned so much from all of your discussions, and Shamil, thank you for moderating. Maybe I'll start by saying that I've been a population geneticist or interested in population genetics since I was 19. It's kind of the only thing I would self-identify as. And my way of thinking about this is that population genetics is just incredibly beautiful framing for understanding evolutionary processes, and sometimes it has application in the outside world, not always, but sometimes it does. And I'm reminded of a quote, just sort of think about a data cloud, think about your favorite, bivariate distribution with a line through it. If you were to ask a biologist, what's real, the biologist would say, well, it's the data. If you were to ask the physicist what's real, they would say, well, it's the line, right? So this has been kind of the tension in the field for a long time, right? Is the model what's real, or is the data and the data is just noise, or is the data what's real, and is the model just, your little toy that you're using to kind of explain the world to yourself? And these sort of meet when we talk about polygenic risk scores and in population genetics, but partly because sometimes population genetics can be very predictive, right? Hardy-Weinberg equilibrium is a really good example, right? That's why we use Hardy-Weinberg equilibrium to QC genotype data. And in fact, in the early days of GWAS, even running a GWAS was used as a QC, right? It used to say the best way to QC your data is go run a GWAS and you'll find all the places where you have bad genotype data. And so today, when we think about polygenic risk scores, one of the problems is that it's just really complicated, right? It's this projection of a really big equation with lots of terms in it, and it's hard to get, some intuition about it. But ultimately, what we really care about, I think, are outliers. So how do you get to outliers in your data, and what do you do with those outliers? And that's a problem that happens all the time in science. So from a clinical point of view, you kind of care about the people who are at highest risk and whether you should treat them differently. And from a therapeutics point of view, you kind of care about why are these people that are at much lower risk, and maybe they've got some interesting biology that you could mirror and drug in some kind of cool way. And for me, maybe what I care about the most is that it's hard to do statistics when you're just extrapolating, especially extrapolating around where you have data. And so to me, the data cloud is here, and I'm interested in all the data that's sort of out here. And so from a very practical point of view, what I've chosen to do is try to go into the real world and see if we can do something about it. So kind of like the UK Biobank. I mean, I love the UK Biobank. I think the UK Biobank is phenomenal. I love Decode. I think Decode's phenomenal. I think, the Finnish stuff is great. It's all great. We just need more of it in other parts of the world. And so what we've started to do is build something we're calling the Biobank of the Americas, because why not? And with the Biobank of the Americas, we'd like to get to about 10 million people in an IRB-approved study, which we've got IRB approval. We unfortunately don't have as much money as either 23 or me or the UK government or the NIH or anything like that. So we got some freezers in Miami. And that's true. But we have CAP accreditation, so we're CAP accredited freezers. And we're taking advantage, or we've taken advantage over the last 18 months or so of the last big surge we had in testing. And it turns out we had a really good opportunity to get a lot of samples because everybody got COVID tested. And one, maybe silver lining around COVID is that it wove its way into all the nooks and crannies of the healthcare system, particularly the understudied population. So it oversampled people at risk, it oversampled poor people, it oversampled minorities. And so we were able to actually get a lot of samples, just literally intercepting COVID samples and getting people to consent to give us their COVID samples. And again, we got kind of lucky because we were able to get about 60 to 70% of people to say, sure, you want to keep my COVID sample and sequence it, that's okay. And so we've been able to collect currently a couple 100,000 samples, but we're on track to try to get to, ideally 10 million samples. And our goal is to maybe use the data first. So again, we don't have as much money as 23andMe, so we can't genotype everybody. But you could sort of maybe prioritize them based on maybe some survey data, maybe some EHR data that can be linked in, and again, sort of look for outliers, sequence them, and come up with a Netflix queue. I like that idea, so I'm going to steal that. So you kind of come up with a Netflix queue of people to sequence, and maybe, down the road you can get pharma or other people to pay for it. Eventually, you want to get to something like a UK Biobank, but for the Americas, and sort of just order of magnitude, the UK is about 65 million people. In the Americas, we have about 650 million people. So if 500,000 people represents 65 million people. It's, you know, kind of double that, and 10 million people should be at least a first place to start to get a representation of the Americas. And I'll tell you one kind of interesting thing that we're doing, which is working with blood banks, particularly in Latin America. And if you work with blood banks, you can actually get access to lots of samples outside of COVID, because, you know, luckily COVID does kind of asymptote. And you can enroll people, and you can get, you know, good usable material. And so we've started to collect that material, and it's not even polygenic risk scores, but just to give you an example, we're doing HLA typing in Latin America, because, you know, we don't actually have bone marrow. If you want to talk about what we don't have bone marrow registry programs in most countries, right? So, you know, so just think about that, you know, that's really actionable information off of a genotype that most people wouldn't, you know, think about whether that's useful or not. So just think about a country like Colombia that doesn't have a bone marrow registry program, but has lots of good donors. And we're now routinely collecting there, and we're also collecting in Miami. Just in the first couple hundred people that we sampled in Colombia, we have already found 4 or 5 new HLA alleles, right? So that's just one example of like beginning to fill in the data cloud so you can begin to build your lines and your models. And on the sampling side, maybe I'll just share from our COVID discards in Miami. My joke is, I'm from Miami, I don't want you to throw the plastic in the water, let's recycle it. We have 87% non-European, just because if you just sit like literally at the Miami arena and give out COVID tests, like 87% of people aren't European. It's not that hard a problem to solve. So we've about a third Hispanic, about a third African American, you know, 20% Asian, and you know, 20% European. So it's pretty good, you know. And the nice thing about having some Europeans is you got to cross-validate. So we cross-validate with the UK Biobank and others. So I do, you know, I'm optimistic. You know, you couldn't be in this field and not be optimistic, particularly with so many smart young people that are doing such great work. So, you know, I'm excited about the future. I do think we need to build better resources. And I don't know whether it'll be better models or new biology, but I do think it's going to have to start with pretty good, big base of samples from which we begin to build. And off of a strong foundation, you can hopefully build stronger and better buildings. So that's kind of my opening salvo, and happy to answer any questions you all may have.
Thank you, Carlos. I really love those bulk data versus outliers ideas. So we have 11 minutes on green. I have no idea how many minutes we have on yellow, but we can move to questions from the panel. Online participants, so please write your questions in the chat. I was told your questions would magically propagate to us. I don't know what's the mechanism, but I assume that this is going to happen. So we'll start from questions in the audience, please.
All right. Hi, I'm Ingrid Holm. I'm at Boston Children's. I'm also part of the Emerge Network, which is looking at polygenic risk scores across the United States. And I guess my question is about clinical outcomes. You know, so we can do all of these kind of studies, but what is, you know, kind of the incremental value of polygenic risk scores when it comes to what a provider says and what a patient does? And it's certainly in Emerge, a lot of the conditions we're doing polygenic risk scores in are conditions where diet and exercise are important, which is, I think everybody knows that they should lose weight and exercise more. Obviously, there's other interventions in terms of, you know, the cancers and cardiovascular disease. But I guess I'm just asking, what do we think, or do you have any ideas about what the incremental value is of polygenic risk scores over and above what's done already? And I guess my second quick question is, as a pediatrician, are people really looked at children? Because it seems to me that if you really wanted to intervene with polygenic risk scores, you do it in babies and parents, because then you could really make a difference in some of these lifestyle things with kind of diet and exercise.
Thank you. That's an excellent question. So Anirudh, I guess it's for you, but maybe others would pitch in.
So for the question regarding going above what we have as tools, both for prediction as well as for enacting change beyond lifestyle and kind of diet type of things. So in cardiovascular disease, for primary prevention, we have a very good clinical risk prediction tool called the Pool-Court equations, where you put in a lot of different clinical variables like blood pressure, cholesterol, smoking, and it spits out a number. And that's an absolute risk score for the chance of getting a heart attack in the next 10 years or a stroke. And one application where polygenic risk scores have been propose could be an added value would be, so when this pooled chord equation spits out an estimate, it can be categorized as low, borderline, intermediate, or high risk categories. And if you're in intermediate or high risk category, then even if, regardless of if you have disease or not, we would recommend starting a statin or cholesterol lowering therapy. So a lot of studies have focused on the people who are in the borderline or intermediate risk category, and then doing polygenic risk assessment in those people, and looking at the reclassification that can occur from being in that low, intermediate or borderline gray zone into truly high or truly low risk categories. And that's a helpful way to kind of introduce polygenic risk scores because it's a smaller percent of the population. It does provide added value in an otherwise gray zone. And at least modeling studies have suggested that it would be cost effective in terms of reclassifying up to like 7% of those individuals into the right category. But as far as like pediatric patients goes, so the pooled chord equation only applies to like age 40 to like 70 something. So for the pediatric cohort, we don't have that. At least I'm not aware of studies that have been done, but one potential application could be similarly to how we do genetic screening, we're using familial hypercholesterolemia variants and doing cascade screening. If someone is found to have a high polygenic risk score, potentially family members who are in the younger age groups could be screened that way. But as far as, I'm not aware of it actually being done.
Thank you. Anybody else, or we move? I think we have to move forward with more questions, so please.
My name is Aaron Abend. I'm Executive Director of the Autoimmune Registry, and I'm looking forward to more work on autoimmune disease because it's the kind of condition that where polygenic risk scores could be a big help in diagnosis. But I'm actually putting on a different hat to ask all of you if you're familiar with the precision medicine program run by the NIH called the All of Us program. Because everyone, not just the panel, but everyone listening should be aware that that program has data on 500,000 Americans. They have EHR data for 200,000 plus of those, as well as survey data and whole genome data for 100,000 of those people. And it's an amazing resource that I urge everyone to look into for doing this kind of research, and I just want to make sure everyone's aware of that. So if you have any questions, feel free to reach out to me, and I'll be happy to answer.
Sorry, was this a comment that in our discussions about amazing data sets we did not mention All of Us, or you actually have a question?
No, I'm just making sure all of the people are aware that the UK Biobank, this is the NIH response to that specifically.
Yeah.
And it doesn't seem like people are using that resource quite enough. And it's very diverse data set, I should also add.
Right. Alicia, would you say a couple of words about your experience with All of Us? Or,
Sure, yeah. All of Us is a phenomenal initiative, and it's, I think, getting off the ground really at a rapid clip now. So I think the genomics data set that has just been released in the past, several months or half a year, something on that order, has been at a sufficient scale to improve polygenic score accuracy for underrepresented populations, but I would say that it's only grown to that scale very recently. So I think the work has just not caught up to where the UK Biobank has been. And additionally, the access to All of Us has been, through this amazing research workbench that democratizes, I think, the access to many, many researchers, only American researchers. So that's been a limitation that I think needs to get sorted out so that the whole world's best and brightest can start to make use of this wonderful resource as we start to carry this forward and ensure that polygenic scores can be more equitably calculated for everybody.
And as a representative of people who analyze the data, I'd like to repeat and kind of underline this point, right? So that bringing infrastructure and collecting amazing data is important, but also sharing and democratizing access. And this is where we should use UK Biobank as an example. Because that's what speeds up development and new methods, new analysis, new results, new scores.
The other thing that I find just super interesting is that the UK is far easier to work with as a company than the US or the US government. I mean, I can get It's easy to get access to UK Biobank. It's easy to download UK genotype data. You can't, the genomics data that you have to do there, partly because just the size. Even Genomics England, which is linked to the clinical data, you can get access to as a private company, whereas the NIH is much more difficult to work with. And then also on the equity side, I would be remiss if I didn't mention the number one question we get is this going into U.S. federal database? Is the U.S. government getting access to my sample? No. Okay, then I'm happy to sign. I'm not kidding. Like it is like people have no problem giving a company access to your data. We do it all the time, right? It is the devil's bargain we make. We're far less comfortable giving the government, and especially minority populations, giving the government access to our materials.
Yeah, just following up on that point a little bit, we, for the UK Biobank, released 12 terabytes worth of summary statistics when we ran GWAS across the entire consortia. All of the, there's 7,000 phenotypes that we ran GWAS on over the course of 48 hours, accumulating something like $80,000 worth of cloud bills, because we're really good at racking up compute costs. But we're able to turn that around and produce a community resource that's a little bit more challenging to do, I think, in the all of us setting. That's not to say it won't get done, it will, but it's going to take longer than 24, 48 hours to do it, to build that infrastructure because of the access challenges.
Yes, I'd like to point, because some
I'm also
people,
gonna
I think this,
one
by the way,
more.
thank you very much for the feedback.
Yeah,
'Cause I'm
I'll
gonna bring
make
it
one.
back.
I mean, as long as we got the bully pulpit.
Yeah.
The Million Vets Project is actually probably the biggest missed opportunity
That's
that
another
we
one,
have
yeah.
in this country, right? Our servicemen and women have, served our country. They deserve, excellent care. We've invested a lot of money. NIH itself doesn't have access to that data.
Yeah.
so
Yeah,
And it is
like-
diverse.
Exactly. It's actually
Well, there aren't a lot
enriched.
of women in it. I do autoimmune disease, so it's got some issues there.
I'd like to maybe again amplify something Alicia said, because some people who are not in the field may not fully appreciate that. So it's not just access to UK Biobank data. So a lot of polygenic scores, a lot of algorithms can be used just on summary statistics, and there are algorithms to test accuracy of your score using summary statistics alone. And summary statistics are publicly available. You don't have to register, no questions asked, released by the Broad Institute one set, released by Brigham and Women's Hospital here another set. You just go online, download it, work with that. So you cannot get back to individuals from this data, but you can train and test your polygenic score method using this data, no questions asked, no registration, no waiting for approval.
There actually have been papers that show that you can use summary statistics to infer individuals, and that's a
But
little
don't tell
scary.
Yaniv early, because he's gonna, if you're
Right.
not Yaniv, it's hard.
Okay, I'm happy to be corrected, but
Yeah.
but they are still released openly.
And it's objectively much harder to do, so
Yeah.
I think the cost benefit there is clear.
Thanks very much for the feedback.
Please.
Hi, I'm Jane Witt. I'm nephrology trainee at Brigham. Sorry for my naive questions about populational genetics, but I wonder, once the sequencing platform is cheaper and we can do full depth whole genome sequencing, do you foresee that depth would change the portability or change things that we see from the SNP-based PRSs?
I'd love to hear your thoughts on this. I have to say, I think working with Latin America, they don't need end-to-end de novo assembly and 30x genomes. They literally need even cheap arrays and HLA typing and panels and things that we know are already working. So there's a really weird gap between what we can deploy, especially at a university, right? Universities get amazing deals on all the technology. They can do it first. It's cheap. It's at Stanford, right? I got free sequencing runs. I was able to sequence every Stanford COVID patient basically for free with Illumina. It was awesome, right? I can't do that outside the United States. They actually charge me much more money to run the same assays outside the United States where they have less resources. So there's this really weird setup where a lot of things end up costing more outside of the US artificially than they need to. And so even if just everybody got Harvard's prices, the world would be a better place. If everybody got the Broad's prices, the world would be a better place. And even for yesterday's technology. So I actually think there's a diffusion, an artificial limit to the diffusion of technology that has more to do with skimming markets than it actually has to do with what's ready for prime time. But
Yeah,
anyway,
Stella, do you have a quick comment, real quick? Yeah.
I think Carl's covered everything.
Okay, so panel members are agreeing with each other. So I thought there would be green and then yellow and red. Truly, there is just red. 5 minutes, yes, sir. Okay. The high authority gives us 5 minutes. Again, online participants, it's 5 minutes. Please go ahead.
So my name is Alok and I'm from Val Cornell. So what are the actually panel's view on the various actually versions of polygenic scores, let's say partition scores are these days where you try to understand the physiology based on the risk, where you divide the polygenic risk scores into the various actually partition of the physiology of disease, or the partition scores just based on the rare variant, but actually partition scores just based on the common variants, or even only on the mosaic actually variants. So how these various actually versions of polygenic scores will help to understand biology, physiology, and diagnostics. So what's the view of panel on that?
I think most efforts in the field treat polygenic risk scores as a prediction tool rather than an etiologic understanding tool. The good polygenic risk scores that explain a large fraction of the trait's variability have many, many variants in it, potentially with very different biological pathways and effects. So there is room for that work to be done, but that's not where we are yet, right? The Kara et al. Paper came out in 2018, and that's what really jump-started this field of polygenic risk scores. So there's a lot of room to experiment with different types of polygenic risk scores, and I look forward to seeing that experimentation. But I think right now we're still in this drinking from the fire hose phase of working with polygenic risk scores. We just want to run bigger GWAS, get higher R-squareds from the models with those polygenic risk scores in them, get the best prediction we can get, hopefully use that predictive knowledge to translate to better health, and additional efforts can then experiment with different kinds and different pathways and different etiologies. But right now, and please, my panelists correct me, it seems much more to confer predictive utility rather than etiologic one.
Like from clinical actually perspective, I have the example of, let's say, type 2 diabetes, where they have the fairly distributed actually partition scores, which helps, like they have list of 716 actually variants, and you can very easily say that somebody have the higher adiposity or higher actually insulin action or actually insulin secretion. That's why the X or Y individual have the high polygenic score. And same goes my own work in Alzheimer's disease now. So I was just kind of trying to understand that, and now with the like maybe trio data or the quad data within the family, how the de novo actually variant will tell you the risk way better than the other actually family members. So that's why I thought like these variants of polygenic score can add much more diagnostic actually value, because actually polygenic scores has been in the field for a while, and one question which I think all of us know as a community, that what's next. And then the second answer we get always is like genetics is not deterministic. So there's the two answers, which is very staple if things don't work. So that's why my perspective. Thank you.
I think it's like, you know, the old adage about a house, but, you know, where you have construction. So I think you can have a polygenic risk score that's easy to calculate, accurate, or useful. Choose two, you know, so.
Yeah. Thank you.
Okay, any more questions? Any more questions from online audience? No. An online question is not identifiable, right? So you can't identify individual based on online questions. So. Okay, so then I'd like to thank all the panel participants. I think this is an important discussion, and thank you all.